Why do we age and what can we do about it? We're in good hands of Professor David Sinclair, who works in the Department of Genetics at Harvard Medical School, and he's also professor in the Department of Physiology and Pharmacology at the University of New South Wales in Australia. He's also co-director of the Paul Glenn Laboratories for the Biological Mechanisms of Aging. During his postdoctoral research at MIT, he co-discovered a cause of aging for yeast, and then was recruited to Harvard Medical School. Dr. Sinclair is co-founder of several biotechnology companies and also a journal called Aging. He has received numerous awards, including the Australian Commonwealth Prize and Bio-Innovator Award. Thank you for coming today, and we can't wait what to do to stop aging.
Thank you, Renata. Well, thank you for coming today. Most of us will only live about 10,000 days, so this is one of them, and hopefully one that's well worth it. Well, so hopefully what myself and, well, thousands of researchers that I represent today will be able to do is to give us a few more days of healthy life. So that's what I want to give you an update on, and where I see the future of healthcare going with advances in the field of aging. Now, there's no question that we live in an incredibly exciting time. I don't need to tell you that the pace of technological change is accelerating. I'm at the point where I feel like my head is spinning off just trying to keep up with it and to utilize this new technology. And I'll just highlight some of the areas that I think are most interesting and relevant to some of my work over at Harvard Medical School. Now, just to give an introduction, though, to ask why is it even worth working on aging, and I do get this question quite a lot, perhaps in a different crowd, though. This is the 20th century version of aging. This is my grandmother, who is now 90 years old, and she lived most of her life in the 20th century, where it was considered pretty good to live to 70 in good health. I don't think that's good enough. I don't think that in the 21st century our children and their children will think of this as good enough. I think they will look back at this time much like we do at the age before antibiotics, with some sort of pity. And why do I think that it's even possible to work on aging, or to do something about it? That's what I'll tell you about today. One of the shocking things that I find about aging today and the way we treat the elderly for lack of technology is that we do put them in nursing homes, and those are the lucky ones, and they end up just aging in place. This is a common term. This is actually just outside the nursing home that my grandmother is currently in Sydney, Australia. And I don't like this term that we just put people away so that they can age in place. And so often I'm asked, well, why work on aging? Well, the goal really is to change the way aging will be experienced in the future. Hopefully it'll be in our lifetimes, and if not, we'll be the last human generation to live a normal lifespan, in my view. So this is how it is for us right now. We spend a large proportion of our lifespan, in general, in a chronically and morbid state, particularly towards the end, but a declining period of morbidity. What the goal with this research is to compress that time of morbidity so that to take it to its extreme, we live to 90 or 100 playing tennis with our great-grandkids, and then we drop dead. Maybe we would spend a few weeks in hospital, but that would certainly be better than what many of us experience and will experience if we don't do something about it. And there's even another option. Even if we're not fully successful at this, even this is preferable, compressing the area of morbidity, even if we don't extend lifespan. So why do I even think this is possible? Well, it's been done for over 75 years now in animals. This is an example of a study out of the University of Wisconsin. Rick Weinbrook is the leader of that. You can see he's aging in himself. I call him the man with the greatest patience of any scientist because he's been doing this for many decades now, running an experiment on monkeys. And you can see, at least from these examples, that if you restrict the calories of primates, and this is true for other species as well, all the way down to budding yeast, you get a compression of morbidity, and you actually do, at least in this study, get an increased lifespan. And this is an example of the data that they've put out, where these are the normal monkeys on a regular chow. This would be pretty much similar to what we would consider a healthy diet that we have. And these monkeys are, these ones are on 20% less calories per day. And this is lifespan if you subtract the non-aging related deaths. And one of the things that's very clear is that cancer is lowered in its incidence, cardiovascular disease is almost wiped out, and there's no evidence of any glucose and metabolic decline. So this is an amazing outcome. We've known about it for a long time, but we can't really do anything useful with this unless we understand what's going on. Certainly, some of us, or some people, not me, but some people are able to do this diet. There's a few thousand people in the world that practice this diet. I tried it for about a week and gave up. It's quite horrible. I wouldn't recommend it, unless you like the feeling of being hungry most of the time. So what we need to do is to understand what's going on and bottle it, essentially. And so basically, eat less leads to more life. What is going on? And this is what my lab and many others have been doing. I've been working on this now for about 15 to 20 years. And actually started here at MIT in the lab of Leonard Guarente over the road here in biology. And what we started working on in the mid-1990s was trying to understand, why do baker's yeast, this is Saccharomyces cerevisiae, why do they grow old? And can we do anything about it? And we very rapidly figured out that they grow old primarily due to genomic instability. The chromosomes recombine and circles of DNA accumulate in these mother cells, which here I'm staining with bromophenol blue. They become quite acidic. These cells actually divide only 25 times. That's their average lifespan. And what we found was that there's a gene that, if you overexpress it, makes them live 30% longer, ostensibly by mimicking this diet calorie restriction. In fact, if you delete the SIR2 gene in this species, calorie restriction doesn't work anymore. And so if you fast forward now 15 years later, we now have a new paradigm of aging, and that is that calorie restriction and other types of biological stress will activate a variety of, let's call them metabolic and stress sensing pathways. We have mTOR, AMP kinase, and this sirtuin pathway that I was just mentioning in yeast, which now we know exists in essentially all life forms, including plants and ourselves. And these are just three of the main pathways that are known to regulate aging in a variety of organisms, and they respond to these stresses and sense changes in metabolites and small molecules. The sirtuins, named after the SIR2 gene in yeast, produce an enzyme that responds to NAD. In fact, NAD, as you'll recall, was originally discovered as a cofactor for redox reactions. We now know that NAD is used as a sensor for metabolism as well as lifespan, and that the levels of NAD decline with aging in many different species, including ourselves, and it leads to a decline in the ability of sirtuins, these NAD-dependent enzymes. These are deacylases in general. They are unable to promote longevity. So how do these pathways promote longevity? Just very briefly, I want to summarize that what we know is one of these sirtuins, called SIRT1 in humans, there are 7 of these in our genome. It has very different roles, but overall protective in many different organs in mammals. And these are the mediators that we know of. These are targets for deacetylation or deacylation. And these are transcription factors in some cases, such as FOXO, NF-kappa B, the inflammatory transcription factor. And really, the summary of this is, I just want to impart that these are protectors of the body, to put it simply, and that they respond to low calories, and now we know even to exercise. And this is a very different view, actually. We used to think that calorie restriction would slow down metabolism, produce fewer free radicals as a result. That's out the window. We used to think that exercise would just pump blood around the cardiovascular system and clean it out. That's no longer valid. What we think is going on is that these are putting a mild stress on the body. We call it hormesis, and that the body responds through sirtuins and these other defense pathways to keep the body healthy. And that's why being lean, dieting, and exercise primarily has health benefits. But you can't ask elderly people to go run a marathon. Many of them are sick, frail. What we need is a small molecule or molecules that you can give to people, perhaps even before they're sick, to not only treat but prevent diseases of aging. Now, the problem with trying to find a small molecule that turns on an enzyme is that there are only a few examples in the whole of pharmaceutical history of allosteric activators, drugs that actually activate enzymatic activity. So we started looking for this. We were certainly looking for more power to turn on this enzyme. The problem is that it's pretty hard to do. And actually, when we started screening for these, it was considered fairly crazy. And I admit that it was rather risky. I thought at a minimum we find inhibitors that would be useful tools. Now, fast forward 10 years later, we now know that you can turn on these sirtuin enzymes with small molecules. This is a crystal structure that's not yet published, but I can tell you that it exists. And this is a human sirtuin model from X-ray crystallography. This is NAD binding here. That's right. This is a peptide. NAD binds up here. And this is one of these activators, which we call STACs, STAC, which stands for sirtuin activating compounds. The first one that got a lot of the press in 2003 was resveratrol. It's found in red wine, so it made a really good story. It linked the potential health benefits of red wine to aging. But we've really come a long way. I want to stress that this was just the beginning. It was a proof of concept, and that work that was done at a local company here called Sotras, just across the road, produced these molecules from high-throughput screening and medchem. Medicinal chemistry produced 4,000 of these, and these are some of the better ones. Then there was a third generation. The company was bought by GlaxoSmithKline, and they continued the effort, and they're now up to fourth generation stacks. And some of these have now been in humans, as I'll show you. We started feeding resveratrol and some of these other molecules to organisms. We fed them to the yeast cells, to worms, to flies, and even to mice. And in every case, we saw improvements in health. These are some of the mice. One of the things that we noticed was that the mice still gained weight as they got older. These are mice on a high-fat diet in this experiment. But when we opened them up, and I should mention this is in collaboration with NIH, particularly the lab of Rafa de Cabos, and what we found was that these mice, even though they had adiposity, they were much healthier. And their gene expression profile matched that of calorie-restricted mice rather than obese ones. And much less inflammation. They didn't get cardiovascular disease or any evidence of those sort of symptoms. We've also been feeding the molecules to mice throughout their lifespan. This is an experiment that's just recently been published in a couple of papers, again with Rafa's help. These mice are held down at NIH. And this is a normal mouse lifespan. You can see the survival starting at all alive and the number of weeks alive. And you can see that both the mean and, to a smaller extent, the maximum are increased statistically significantly with a couple of these molecules that were made at Sotras. This is 1720, which has also been shown to improve metabolism, prevent cardiovascular disease, at least in mice. This one here is particularly interesting because it's gone into humans. And I'll show you a result from that in a minute. So this is the first example that you can design molecules that prevent diseases of aging and extend lifespan, at least in a mouse. We don't know about humans yet. They don't seem to be toxic, that's for sure, at least in animals. And so far in humans, I have to say that there's nothing that's shown up in preclinical and clinical studies that gives us pause for toxicity. So what are the uses of these molecules? Well, there's plenty of uses. You could pretty much, the problem is there's too many potential uses. Some of the areas that have been considered, some of these have actually been moved into clinical trials. We've got muscle disorders, metabolic diseases, even neurodegenerative diseases, eye diseases. These are all where positive results have been found in mice. COPD, for example, has been quite responsive to these stacks. And in general, inflammation is suppressed by these stacks, and that's a hallmark of calorie restriction. So one of the diseases that is most advanced is this one, psoriasis. This is, I'm sure you're aware, is a horrible disease for those who have it. It's a inflammation of the skin. You end up with a number of inflammation spots. There's a particular form of this called plaque-type psoriasis, which I'm showing you here. This is an example of a patient before treatment with the molecule 2104 that I showed you in mice, extends lifespan. This is the front and back of the legs of this particular patient. I should give credit to Jim Kruger at Rockefeller University for conducting this study. So this is an oral drug. It's given just as a pill. Right now, the current treatment for plaque-type psoriasis is injections with anti-TNF antibodies, which is the patients somewhat respond. It's not a bad drug, but an injectable, of course, is not ideal. If you could just take a pill, it would be great. And there were significant improvements in most of these patients. You can see here the same patient. This is after 40 days treatment with this oral developmental compound. So where we stand now is that we know, at least in mice, it's pretty easy to extend lifespan and slow down diseases of aging. You can even use some of these to, at least in early studies, treat some diseases that are associated with inflammation, possibly aging. And I just want to, in the last few moments that I have, touch upon some other high points that I wanted to tell you about, new frontiers that I'm aware of and some of which I'm working on. One of them is that it's a real problem doing one of these mouse studies. They take a few years. So what we'd like to do is accelerate the ability to discover new drugs in mammals, certainly test them. And what we've done over at my lab at Harvard is generate a mouse that we believe is the closest mimic of normal aging. These are both the same age. They're both, in this photograph, 8 months of age. And this is the control mouse, and this is its sister here. And this mouse is an inducible aging model. We feed the mice with tamoxifen, and what we induce are epigenetic changes that we believe underlie aspects of aging in mammals. This is an unpublished mouse, but we're now characterizing this both at the phenotypic level and the genetic level, the molecular level, and trying to understand what are the epigenetic changes that we've induced in this mouse. You can ask me later if you're interested more about how this works. But we think this mouse, which ages in just a matter of months, can be used to more rapidly test some of these compounds that may be useful against aging. There are already people who live seemingly with resistance to diseases of aging. This is an example of a couple of guys in the same family in New York. These are neobarzilai's patients down in New York. Typically, he looks for Ashkenazi Jew families that live longer. And this family tends to live over 100. You can see this gentleman here looking pretty good for 100. And in general, Near tells me that offspring who are from centenarian families not only are resistant to diseases, but they tend to look better as well. And I do agree, for 70, this man's looking pretty good. So there's now a large effort from a number of academics and now companies that have recently formed to sequence whole genomes of these individuals, as well as look at their gene expression, as well as their DNA methylome, for example, to try and understand what is different about these individuals versus the rest of us. We have some idea that through GWAS analysis, we know that there are some variants, but in general, so far, it's surprising how little these individuals differ from us. And so that's why I think we need more depth of analysis. They also don't have a healthier lifespan. A lot of the centenarians drink and smoke heavily. So there's something different. We just don't know exactly what that is yet. I did want to point out that there's a paper that came out fairly recently that associates variations in the SIRT1 gene that I was talking about earlier with longevity. And this is just it's early days, but it may be that this gene is involved in human longevity. There are plenty of studies actually now that show that variations in SIRT1 and its promoter predispose or protect you from cardiovascular disease and also diabetes type 2. So this is another area that I think I'm not alone in being excited about. So what do you do if you're born with one of these variants that predisposes you to a disease? Well, what we're working on with George Church over in my department in genetics is seeing if we can edit our genome. We believe it's going to be important in the future to be able to change the way our organs behave, as well as, excitingly, be able to change the genome of future generations. And I know that that's controversial. What we're working on now is the ability to correct genetic diseases in the germline. And what makes that feasible is that we have what we believe are stem cells that come from women's ovaries. We can grow these in the dish and they pop out eggs here in culture. This is a courtesy Over Science, a company that, in full disclosure, I co-founded a couple of years ago. And these cells actually can be induced in co-culture to turn into fertilizable eggs. And these have been used in mice to generate offspring, genetically altered offspring. But these are actually human cells that you can isolate pretty easily through faxing, sorting of cells from women. And what we're going to do, and in the middle of doing, is taking, first of all in mice with, let's say, genetic models of disease, such as Huntington's and BRCA1 mutation for breast cancer susceptibility, and we're able to use this technology, CRISPR, to go into the genome and create clones of these stem cells, and then sequence these to make sure that we've just altered the right base pair, and then use those clones to generate offspring, and that these offspring should be genetically corrected. And then we are in the midst of following that shortly up behind with doing this for humans, and this would be an IVF procedure. So I think that it's an extremely exciting time for us and for our children. This is my son Benjamin, who was, at least before the FDA shut them down, was able to do a 23andMe test and have a look at his variants, and if nothing else, learn about genetics and the exciting future that his generation can look forward to. And I have a lot of people to thank for the work that I presented today. They're up here. And again, thanks for inviting me.